As dear children, CO2, the present topic and experiment is CO2 is necessary for photosynthesis. The necessary means very important. Without that, photosynthesis cannot happen. So this experiment was done by a scientist called Mohl. The scientist name is Mohl. It is also called Mohl's experiment or Mohl's half-leaf experiment. Mohl's half-leaf experiment. Why it is called half-leaf experiment? It is named with a scientist name called Mohl, so called Mohl's experiment. And why it is called half-leaf? I will show the textbook page to you. This is a diagram which shows carbon dioxide is essential for photosynthesis experiment, which is also called as Mohl's half-leaf experiment. You can see its title down, Mohl's half-leaf experiment, because here in this a leaf half part is kept inside the bottle and half part is kept outside. So a leaf is kept with half zone with one experiment and half zone of the leaf with another experiment. So as the leaf is separated with two halves to do different activities, so the experiment is named with Mohl's half-leaf experiment. Let us see its procedure of doing. So here Mohl had taken a kind of plant, a potted plant. Which type of potted plant? A potted plant with long narrow leaves. So it is not a normal plant like others. The plant should have long narrow leaf. Now the question is, why to take long narrow leaf containing plant? The answer is the experiment is half leaf experiment. If the leaf is like this with a normal shape, can it be inserted into the bottle? No, it should be narrow. And if it is short, can half be inside and half be outside? No. So to make a possible arrangement in this procedure, in this experiment, to make a possible arrangement, a long narrow leaf containing a plant is selected by Mohl. So it is possible to keep in bottle the half of leaf easily into the bottle and half of the leaf is easily outside and visible and will be useful for the experiment. So first thing is he taken a long narrow leaf containing plant, and then he kept this plant in dark room without the availability of sunlight around for 3 to 4 days. 3 to 4 days. Again a question, why? Second question. This is all cares he is taking before doing this experiment called Mohl's half-leaf experiment to know the importance of carbon dioxide. So he kept this plant in dark room for 3 to 4 days. And coming to the reason why. What happens if a plant kept in sunlight? It will perform photosynthesis and it will prepare starch. And what happens, the same plant is kept in dark room for 3 to 4 days. No sunlight availability, no photosynthesis, no glucose preparation, no starch storage. So there will be no starch. Complete starch will be removed from the leaves of a plant which is kept in the dark room. Is it or not? So if we supply the food continuously for every 3 to 4 hours, the food will be stored inside. Just like glucose stored inside a leaf as a starch. If we stop taking food for some days, the reserve food will be used by us and we become without reserve food in the body, in the cells. In the same way, when a plant is kept continuously in sunlight, it produces glucose and stores starch and reserve it. But if we keep it in the dark room for 3 to 4 days, then the plant or leaf becomes without starch, without reserve food called starch, and such a plant is called de-starched plant. De. If you add anything called de in front of anything, that is called removal. Without. Deactive, active, deactive. Dehydration, hydration, taking water, dehydration, removal of water. So there are some technical rules. Re, if you add re means again, regain. Understand to you? Rehydration, repeated. Understand? If you add re in front of any word, it means recycled. It is done again. But here the word is de-starch. De means removed. Removal of starch by keeping in dark room for 3 to 4 days. So again a question, why to de-starch this plant? Why to remove the starch from this plant? The answer is, before knowing the importance of carbon dioxide in preparing starch, you are telling that carbon dioxide is necessary for photosynthesis to prepare starch. If starch remains in this leaf, if starch remains while we are doing experiment in this leaf, we fail that new starch is formed or not, because already starch is there if you not de-starch any plant. So if you not de-starch a plant before doing experiment, some starch will remain in the leaves. And if you test for starch, we don't know it is newly prepared starch or already existing starch. So before knowing about starch is prepared by plant or not, we should remove the starch. Whatever previously present, whatever reserve already in the leaf should be removed. Then only if we start doing experiment, we will come to know that whether new starch is prepared or not. So not to interfere the experiment, not to disrupt the experiment, not to give a negative result in the experiment, we removed the past or reserved starch present in the leaf by keeping it in the dark room for 3 to 4 days, and the process is called de-starching. It's a very important question. How a plant can be de-starched? Simply keeping it in dark room for 3 to 4 days. By keeping a plant in 3 to 4 days in a dark room, what happens? It is de-starched. So understand it. Why a plant is de-starched before doing an experiment for photosynthesis? To remove the reserve starch before doing experiment, to know whether new starch is prepared or not. Understand to you? So we need to find out the new starch is prepared here or not. So we removed the complete starch from the leaf by process called de-starch. Now the experiment procedure can be started. So if a question comes, what care taken in this experiment before starting this Mohl soft leaf experiment? Taking a kind of special plant, that is plant with long narrow leaves, and de-starching the plant not to interfere, not to trouble the starch while doing experiment. So not to interfere the starch, we done de-starch. De-starching of plant is very important. So let us start what arrangements, what apparatus he taken further to continue this experiment.